Hello and welcome. Today we're going to be talking about nucleic acid biochemistry in today's lecture. And so we're going to cover the macromolecules as well as their monomers and the unique biochemistry of these components that allow us to have a little bit more control over our environment and our genetic code. So we're going to get started off here today. We're going to be talking about some nucleic acid biochemistry that has allowed us to take control over some serious diseases. When you think of nucleic acid biochemistry, I want you to think about pandemics, recent and more distantly in the past. So what we're looking at here is a picture of the NAMES Project AIDS Memorial Quilt. Covers almost 20 acres, over 54 tons combined weight. And the weight is nothing in comparison to the gravity, because each panel is roughly the size of a grave, representing one of the 96,000 people who had died by the time this project began. They died while the scientific and governmental communities around the world languished and failed to produce or act on providing a treatment for HIV/AIDS. The quilt itself is housed not too far from you where you're located in Oakland. It's in San Francisco. You're looking at a picture of where it's displayed before the Washington Monument in Washington, D.C. Since 1981, over 40 million people have died of AIDS. Currently, however, there are 38,400,000 people living with HIV infection, about 75% of which are receiving antiretroviral treatment. Today, when we're talking about nucleic acid biochemistry, I want to talk to you about some of the things that have allowed us to reduce the death rate of HIV/AIDS by 50% in the last decade. So let's go ahead and get started on today, the 28th of February, 2023. What I did is I took the chapters in your book about nucleic acid biochemistry and I created a word cloud out of them. And so the biggest words were the ones that came up the most frequently. So we've got big words like temperature and hydrogen bond. And we know from previous lectures that hydrogen bonds are susceptible to temperature. They form at lower temperatures and they break apart at higher temperatures. That's important for nucleic acid biochemistry. We've got mitochondrial DNA. Mitochondria have their own DNA. We've got nucleus and cytosol. Genetic DNA in eukaryotes is housed in the nucleus. RNA is transcribed and then moved, transcribed in the nucleus and then moved to the cytoplasm. Some of these components of nucleic acids are themselves used as neurotransmitters. There's some different sugars like ribose and deoxyribose. There's some big names on this word cloud as well. Rosalind Franklin, Elizabeth Blackburn, Barbara McClintock. We've got quite a lot of large shoulders that we stand on. Enzymes, nitrogenous bases, we'll talk about a lot of these different things. And so hopefully this will get us focused for the lecture. So life started with nucleic acids. All of Darwin's endless forms most beautiful arose from the chemical properties of nucleic acids. And these chemicals had to self-organize under prebiotic conditions. That is, before enzymes or anything that could carry out replication or transcription or translation or any of the processes that create these, they had to self-organize. And part of that is due to the unique chemistry that they have. And we'll talk a little bit about that later. And evolution from that point evolved so far into the future that these genes, these sequences of nucleic acids, were able to invent the very tools that we now harness today to potentially make even more endlessly beautiful creatures. This, of course, is CRISPR, which you've watched a video about, and there's a whole slew of ethics that we can talk about with the use of CRISPR as well. But the point is that we have evolved beyond just studying our genes and our nucleic acid structures. We are now capable of using that information to cure disease. And we'll talk a little bit about that. So nucleic acids, in the grand scheme of things, fall into a regimen here. We've got DNA, which, as I mentioned earlier, needs to replicate itself, and this is a process we call replication. In eukaryotes, this happens in the nucleus. In prokaryotes, this happens in the cytoplasm. Then it is transcribed to RNA, and then from RNA it is translated to protein. And from protein, the protein folds up into a structure of some kind, and that structure has some kind of function, and that structure is transduced into a function. And that function plays some role in the environment. It either makes the organism more fit or doesn't play any role to do with fitness, or it makes it less fit. And so the role of the function in this scenario here is to feed back to the DNA the functions of proteins that ultimately make an organism more successful in its environment, will allow it to propagate its DNA through time. And we call this selection, where the environment selects for DNA sequences that are handy at sticking around. They meet the demands of the natural landscape. And then the ones that the sequences that do not perform well drown in the gene pool. Most of these arrows point in a forward direction. There's one notable exception here, which is from RNA to DNA, a process we call reverse transcription, very cleverly. And also, fun fact, there's a little bit of replication in the eukaryotic genome that is also considered by default reverse transcription. So all of these processes are necessary to carry out life as we know it. And we call this relationship between macromolecules the central dogma. Now, dogma is a term that means something that is generally assumed to be true. For a long time, we thought the arrows only went in the forward direction, and then we discovered reverse transcriptases. And so as scientists, we are open to the idea that there could be other arrangements here, but for the most part, this is the primary understanding we have about the relationship between these macromolecules, the relationship between a gene, its transcribed RNA, and the protein and the functions it may produce. So let's take a look at DNA and RNA. These are our major macromolecules of the nucleic acids, and they have pretty different structures here. So DNA, you'll notice, has two strands that form a double helix, whereas RNA is a little bit more willy-nilly, topsy-turvy. Sometimes it's single-stranded, sometimes it binds to itself. Not exactly a regular structure. DNA, on the other hand, has a much more regular structure. This is, looking at these structures, this is a way that you can remember which nucleic acid does what. So the very regular structure of DNA makes it very well suited to being the long-term storage information, and the very malleable, willy-nilly form of RNA makes it more amenable to being kind of like a scratch pad, like a molecular sticky note that copies down information from RNA and then is used as blueprints for protein. Okay, so let's take a look at the individual building blocks that compose these nucleic acids. So nucleic acids are composed of nucleotides. So this first one here is called adenosine, and this next one here is called guanosine. After that, we have what's called cytidine. And then next up, we have thymidine. You can see their one-letter codes are coming up. And then lastly, we have uridine. And technically, these are all the triphosphate derivatives of these molecules. So it'd be adenosine triphosphate, guanosine triphosphate, cytidine triphosphate, thymidine, and uracil triphosphate. These molecules, when you look at them with our eyes at these kind of space-filling and wireframe models of the molecules, it might be difficult to our eyes to make obvious differences as to what these, how these chemicals differ. But make no mistake, the enzymes that go on and carry out the processes of replication and transcription and translation, They are very specific. They can tell the difference between these molecules. And we'll get into a little bit how they do that. But generally for now, we can break these 5 molecules up into 2 distinct categories. We have purines and pyrimidines. So adenosine and guanosine are the purines. And I like to remember this with big base, little name. So the purines have 2 cycles on them. They have two rings attached to the sugar and the phosphate. And the pyrimidines have just 1 ring attached to the sugar and the phosphate. So cytidine, thymidine, and uridine are all pyrimidines. They have 1 cycle and a big name. So purines, big base, little name. Pyrimidines, little base, big name. Let's take a look here just at adenine for a second. Excuse me, adenosine. And we're going to use this as a template to understanding what goes in to a nucleotide. Like, so we've just broken the nucleic acid down into nucleotides, and now we're going to break the nucleotide down into its chemical components. So first off, we have what's called a nitrogenous base. And this is what defines the name of each of the nucleotides. So adenosine has adenine, guanosine has guanine, cytidine has cytosine, thymidine has thymine, and uridine has uracil. So each of those are molecules that you need to be aware of, and generally what their structures are and who they bond to. We'll get to that later. But the nitrogenous base is a definitive component of a nucleotide. Everything else is going to be relatively standard across the nucleotide. So you've got this nitrogenous base, which is bound to a sugar, which is in turn bound to a phosphate. And it's always going to be in this order. You're going to have phosphate, sugar, nitrogenous base, sugar, phosphate. You're not going to have phosphate in the middle. You're not going to have a nitrogenous base in the middle. It's always in this order. Phosphate's on the outside, sugar's in the middle, nitrogenous base is on the other side. Okay. The sugar is the key sweet ingredient in this, right? So let's refer back to our sugar biochemistry that we learned early in the semester. We've got D-ribose here, and we've got that first carbon molecule, carbon atom. We call it the first carbon atom because in the straight chain, remember, that carbon used to be an aldehyde, but double bound to an oxygen. So it has the highest priority when you do your naming conventions. And that, that gives us the number 1 carbon there. And then we can see that we have a number 2 carbon that's adjacent, number 3, number 4, and number 5. And so we'll refer throughout the lecture to hydrogens and hydroxyl groups that are attached to these carbons. So this is the 3' hydroxyl. This is the 5' O hydroxyl group that is in turn replaced with the phosphate. So looking at this here, we've got D-ribose in this configuration, and we're going to attach it to a nitrogenous base. And so we'll start off by making the chemical we have before us, adenosine. We'll start by bringing adenine in. And I'm going to show you, because it's important for how polymerization works, I'm going to show you how the molecular mechanism by which all the electrons move around and create this very important bond between the sugar and the nitrogenous base. So those 2 electrons on adenine's number 1 nitrogen come in and they attack the number 1 carbon, which pushes electrons up onto that oxygen, which pushes some electrons onto a source of acid in the environment. And so that is a hydronium ion. That's basically just a water molecule with an extra hydrogen group attached to it. And this exists in some amount throughout the, even in normal pH 7 water. Some amount of this is always going to be present, even if just for a microsecond or so. And so then what happens there is when that oxygen molecule, those electrons attack that hydrogen, it pulls the hydrogen off the hydronium, and 2 water molecules leave, and that leaves behind this bond to the number 1 nitrogen on the adenine purine molecule. But that molecule also has a positive charge because now nitrogen has too many bonds and not enough lone pairs. So a water molecule comes in from the environment. It will abscond that hydrogen right off of it because it's primed and ready to go. That nitrogen has its hands full and it's ready to let go of that hydrogen. Any nearby water molecule will come by, and then that hydronium molecule is reformed. So we say that this is an acid catalyzed reaction, and it leaves behind what's called a beta glycosidic bond. Now we call it a dehydration reaction because water was the main chemical that left. Okay, while it was acid catalyzed via that hydronium, water, H2O, was the molecule that ultimately left. And so this dehydration reaction is a major component of the polymerization that we're going to see in DNA, attaching nitrogenous bases to their sugars and then attaching one nucleotide to another nucleotide. This is also the reaction that allows for the polymerization of peptides. So a peptide bond is formed by a dehydration reaction. And so it's that removing of water. Anytime you see a hydroxyl group, I want you to think of it as a place that you can rip off hydroxyl and pull two molecules together. Okay, so that beta glycosidic bond is what holds the nitrogenous base to the sugar. And when you add a phosphate group to that sugar, then we give it this name, adenosine triphosphate. In this case, because there's three phosphate groups, there's we say triphosphate. Two phosphate groups, that'd be diphosphate. One phosphate group, that'd be monophosphate. And so the name of this molecule is named after the nucleoside, which would be nucleosides are the nitrogenous base and a sugar. And then if you add any phosphates to a nucleoside, you call it a nucleotide. And so this is adenosine triphosphate. Now this molecule is very important. We're very familiar with this. ATP is used as a source of chemical energy. It's that energy held in those phosphate bonds is useful to do molecular work. And it's also a component in acetyl-CoA, which is an enzyme, a coenzyme factor that allows for enzymatic reactions to take place. If you take this first, if you rip off these two phosphate groups here and you rip off this, or you take this phosphate group and bend it around to this hydroxyl group, you can create kind of a kinky little loop here, and then that was what we would call cyclic adenosine monophosphate. So one phosphate forms a cycle back onto the ribose, and it would have the adenine hanging off by that beta glycosidic bond. That would be CAMP, cyclic adenosine monophosphate, and that's an extremely important second messenger signal. And of course, ATP is a component of RNA, one of the nucleic acids we're talking about today. But DNA, on the other hand, is not made out of adenosine triphosphate. It is instead made out of deoxyadenosine triphosphate. Notice here the 2' hydroxyl is missing. It would be replaced by a hydrogen group right here, a hydrogen atom here on the 2' carbon of deoxyadenosine. Deoxy just means we took off the oxygen, we took off the hydroxyl group that contained that oxygen. And so deoxyadenosine triphosphate is used for DNA. It's important that your long-term storage molecule, DNA, long-term information storage, can't be mistaken for energy. So that's one of the reasons that you might want to, one of the reasons that you might want to have two separate molecular classes for your long-term storage and your short-term storage that might also double as chemical energy and second messengers and things like that. So let's talk a little bit more about why these sugars matter. So what you've got here in blue is a DNA polymerase, then you've got the RNA chain here, and then at the end is an adenosine triphosphate, not deoxy. Let me go ahead and rip off that hydroxyl group. That's what's blinking red on and off here. That hydroxyl group is the difference between deoxy and normal adenosine triphosphate. And you can see that this tyrosine group here, if we turn that into a space filling model, we can see that there's just no space there whatsoever for another hydroxyl group. And so when I say that these sugars matter, I mean it matters to the enzymes that are going to be dealing with these molecules. So RNA is going to be made by RNA polymerases that are specifically tuned to be specific for nucleotides, whereas DNA is going to be made by polymerases, enzymes that are specific for deoxynucleotides. They are going to have specific functional structures that recognize their molecular, the correct molecular monomer to grab onto. So when we talk about the structures of these molecules, to us, it's just a kind of a cartoon on the screen. To the molecules, it either fits in like a Lego or it doesn't, right? It either meets the demands of the biophysical active site or it does not. And so that's why these sugars matter so much, is because the enzymes that use them will either recognize one kind or another. So we just talked about putting nitrogenous bases onto the sugars. Let's try now to connect nitrogenous or nucleotides to one another. So this squiggly line up here means that there's some RNA chain above this, and that we're looking at the last two nucleotides in a long RNA chain. We've got an A, we've got a G and a C, a guanosine and a cytidine, and we're going to take an adenosine triphosphate and we're going to add it to the growing end of this chain. We're going to elongate here. So now what you're looking at here looks kind of messy. There's a lot going on here. There's negative charges, lots of polar molecules in here. This is relatively unlikely to be neutral in the environment. And unlikely to happen because of all the confounding factors here. You've got these negative charges and these polar things that would have partial negative charges, and so on the whole, this there might be some competition between molecular forces here. Well, don't worry, that's what enzymes are for. So we're going to have in the background here, we've got this large RNA polymerase molecule that is coordinating this reaction. So it's going to be holding on to the chain that it's growing. It's going to look for the incoming nucleotide triphosphate, and it will hold them all in the right position so that this reaction, which is also a dehydration reaction, can form. So this will rip off two phosphates that water leaves away, leaving behind what's called a phosphodiester bond. And again, that was formed by a dehydration reaction. So when you add adenine to, or you add a purine or a pyrimidine to a ribose sugar, that is forming a beta glycosidic bond, and the process that made it is called a dehydration reaction. When you add two nucleotides together via a phosphodiester bond, that phosphodiester bond is made by a dehydration reaction here. And so that was all catalyzed by the RNA polymerase in the background. So that's the process we call polymerizing. Now, as we know, DNA and RNA can bind to themselves and to each other as well. So let's look at how that happens. So in the opposite direction, we're going to have a molecule come in and bind on the other side of this molecular ladder here. So you've got guanosine binding to cytidine via 3 hydrogen bonds. And so I've got hydrogen bonds depicted here as like an electronegative side and an electropositive side. You can see this nitrogen group is sucking all the electrons away from this hydrogen, which makes it electropositive, and you've got all these electron densities around the double bound molecular oxygen here, and those two partial charges, partial negative and partial positive, those form an attraction. And cytosine and guanosine, cytidine and guanosine are attracted to each other in this way. For simplicity's sake, we can refer to these bases by by their bases. So this would be cytosine and this would be guanine, and that is that kind of nomenclature helps when you're trying to talk about things that may be RNA or maybe DNA. You can refer to them by their bases. But for now, I'll refer to these as cytidine because it holds on to that 2' hydroxyl, and guanosine because it holds on to that 2' hydroxyl. So you've got these 3 hydrogen bonds here. And now you can imagine that if C binds to G here, then C is going to bind to G here as well. So we've got those same 3 hydrogen bonds just on the opposite side. And then in RNA, adenosine binds to uridine, and it does so by 2 hydrogen bonds, which of course means that the connection between adenosine and uridine is just a little bit weaker. But they still are, it's still based on, gradients of electropositivity to electronegativity. Now, if this were DNA instead of RNA, it would look like this. So this would be deoxyguanosine, deoxycytidine, deoxyadenosine, and deoxythymidine. This is 2 strands of DNA that are complementarily bound to one another via base pairs. Now you'll notice that in DNA, adenosine and deoxythymidine, they bind to each other in a very similar pattern, and that is by design. So thymidine is the DNA synonym for uridine, and in RNA, uracil or uridine is the synonym for thymidine. Now, again, I want to point out some structural differences here. We've got hydrogen bond or hydrogen at the 2' of each of these sugars instead of the hydroxyl group. That goes back to the structure of that molecule, the RNA polymerase in particular. It's only going to recognize this because if these were all hydroxyl groups, they'd be too bulky and they wouldn't fit in the polymerase active site like we talked about earlier. Another difference here is that thymidine and uracil are very similar in structure. If we observe here, there's just a methyl group that adds on there and differentiates thymidine from uracil. That methyl group can make it more, it makes this double bond in particular a little bit more stable, and it may cause problems elsewhere, but we'll talk about that later when we talk about sunscreen. So some key features of DNA. It is, the main differences are that there's no hydroxyl group at the 2' location, and it uses deoxythymidine instead of uridine or any form of uracil. We say that its structure is anti-parallel, and what that means is that if you align all of these sugars up like this, we see that this is the 5', the 4', the 3' carbon, 5', 4', 3'. In the other direction, we have 5', 4', 3' running anti-parallel. They run along the same line, but in opposite directions. And so we can see that visualized here. So DNA, especially the double helix, is anti-parallel, and each, purines are always going to bind to pyrimidines. Pyrimidines are always going to bind to purines, and in this way, these are what we call base pairs: guanosine to cytidine, adenine to thymine. Those are our base pairs. It's T not U. That's another major difference. No 2' hydroxyl. I can't stress that enough. DNA forms a double helix, and that double helix is not perfectly cylindrical or perfectly regular. There's a major groove and there's a minor groove. The major groove is where most proteins interact with DNA because there's just physically more space for molecules to interact with the nitrogenous bases. There's a negative phosphate backbone running along the rungs of DNA and RNA. The nucleotides are held to one each other via phosphodiester bonds. Ester, of course, is just a connection between molecules where oxygen is the intermediate. So we've got carbon, oxygen, phosphate, oxygen, carbon. And so that's 2 ester bonds and a phosphate molecule or atom in there. So we call that phosphodiester bonds. Hydrophobic core. Now, let's point out that everything here in the middle of the helix, it's not nearly as hydrophobic as, say, a lipid by any means, but there is a gradient of hydrophobicity. The outside pore, the outside of the helix is going to have these negatively charged phosphate groups and these polar hydroxyl groups hanging off of the sugars. But in the middle, there's going to be a relative increase in hydrophobicity because you've got these double bonds, you've got these carbon groups, and at least charge-wise, a lot of the partial charges are balanced out by these hydrogen bonds. And so on the whole, the inside of the helix is a lot more hydrophobic than the outside. But again, this is all relative because the inside of DNA is nowhere near as hydrophobic as, say, a lipid, but that property does influence the kinds and shapes of proteins and the chemical properties of those proteins that would interact with DNA, right? If they were going to interact with the phosphate backbone, they would have to be positively charged to attract to it. If they were going to interact with the major groove in these nitrogenous bases, it would probably want to be a little bit more hydrophobic, not something supercharged. And then, of course, there's always case-by-case bases there. And then we've got a hydrophilic surface. Now, DNA denatures with sufficient temperature. And the reason is because these hydrogen bonds are sensitive to temperature. So if we raise the temperature on this strand of DNA, those hydrogen bonds will denature and the molecule will separate out from itself. And this is a major key feature of the structure of these nucleic acids that's important for several technologies that we use today, like sequencing as well as polymerase chain reaction PCR. And so we use temperature in the lab all the time to rip DNA strands apart and get access to the information that's held in these hydrogen bonds. Now let's take a second here to just take a beat and clarify some nomenclature. You've got a nitrogenous base, and that there are 5 of them. Only 4 exist in DNA, only 4 exist in RNA, but there are 5 of them. T and U are swap out for one another. You've got adenine, guanine, cytosine, thymine, and uracil. And once you attach those to a sugar via a beta N1 glycosidic bond like this, then it becomes a nucleoside. So the nitrogenous base added onto a sugar is called a nucleoside. And then they suddenly have different names. So adenosine, guanosine, cytidine, thymidine, as opposed to cytosine and thymine, and then uridine as opposed to uracil, right? So their names change when their structures change. Something I want you to be aware of, though, is that because there's 5 nitrogenous bases, but some only exist in RNA and some only exist in DNA, for the purposes of my course, when I talk about adenosine, I'm talking about a molecule that goes in RNA. If I'm talking about deoxyadenosine, I'm talking about a molecule that goes in DNA. Likewise, deoxythymidine is the only molecule I care about because it is only used in DNA. I don't care about thymidine because it doesn't end up incorporated into RNA. Uridine, I only care about because it only ends up in RNA. Deoxyuridine doesn't exist in sufficient amounts and doesn't end up in the structure of DNA. And so for my class, you've got deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. You've got adenosine, guanosine, cytidine, and uridine. There's no deoxyuridine and there's no thymidine without that deoxy on it, okay? Second, you add any kind of phosphate group to these nucleosides become nucleotides that are primed and ready to add to a growing chain of molecules, or in the case of an ATP or GTP, to be used for energy or second messaging. And then, of course, they'll end up in DNA. DNA and RNA are the polymers, whereas nitrogenous bases, nucleosides, and nucleotides are the monomers. So let's go ahead and hop over to Menti for a second, Mentimeter, and you can start answering the first question, and we'll see what you've got here. So what are the 5 elements found in nucleic acids? We've got some answers here: carbon, oxygen, hydrogen, nitrogen, and phosphorus. And that seems to be the consensus here. That is true. Oxygen is one atom that we see. We've got an answer down here about sulfur being a component of this. Not sulfur. The missing one from there is phosphate. Phosphorus is the atom, the element that's missing from this. It forms a component of the molecule phosphate that is used for those phosphodiester bonds. Funny enough, you can take nucleic acid structures, nucleotide structures, and substitute out one of those carbons for a sulfur, and you will create a compound that can be used as a drug. We'll talk about that later in the lecture, the use of that kind of strict organic chemistry there. So great, let's go ahead and move on to the next question. I see some of you already answered that. So what molecular mechanism conjugates or attaches nitrogenous bases to sugars and conjugates nucleotides to one another? So this, it looks like, we had a little bit of confusion here. So I used the beta glycosidic linkage as an example of a molecular mechanism that's called the dehydration reaction. But it's very important that we understand that the phosphodiester bond is also formed by a dehydration reaction. So the things that the beta glycosidic linkage and the phosphodiester bond have in common is that they are both formed by a dehydration reaction. That's the mechanism that attaches nitrogenous bases to sugars via a beta glycosidic linkage and nucleotides to one another via a phosphodiester bond. So I see some of you have also moved on to answer your next question. We've got what type of bonds anneal both chains of the double helix. So this is not how are the monomers bound to each other. This is not how the atoms align. This is how does one strand bind to the other. And so we had one for glycosidic linkages. Remember, that's the nitrogenous base to the sugar. The majority, the consensus here was for hydrogen bonds, and that's absolutely correct. And hydrogen bonds do not share electrons, which is the definition of a covalent bond. And so hydrogen bonds only work by having gradients of electronegativity. You've got oxygen and nitrogen, which are electronegative. And if a hydrogen is attached to a hydrogen or an oxygen, those bigger atoms are going to suck away all the electrons and leave that hydrogen to be very exposed and electropositive, and it's going to have a partial positive charge that's going to interact with a partial negative charge on an electronegative atom, and they're going to form kind of a transient bond known as a hydrogen bond that can be overcome with heat. And then lastly, we have this mid-lecture question: a nucleotide has what 3 components? So we've got phosphate, nitrogenous base, and sugar, correct? Quick note here, ATP is a nucleotide. It's going to be composed of a nitrogenous base and a sugar. Not sure, thank you for the honesty. Hopefully this clears it up for you. I like this answer in particular that says phosphate hyphen sugar hyphen nitrogenous base, because I stressed it will always come in that order. You'll have phosphate, sugar, nitrogenous base. Okay, great job. So with that, let's go ahead and move on to, we're going to talk about some big picture things. We're going to talk about the timeline of genetics, as well as we're going to talk about the macromolecules that these nucleotides make up, okay? So starting in 1856, Mendel, a monk, demonstrated that traits are heritable. He was working with pea plants that showed that some characters or traits or phenotypes of the pea could be passed on to the next generation with careful mixing of the genetic material, the pollen and such. A few years later, in 1859, Charles Darwin proposed that traits descend with modification. This is the idea that traits over time are going to become better and better suited to their environment via a process of natural selection. This is the idea that environmental conditions are going to allow some traits to survive on, and others will drown in the gene pool. Funny enough, both Mendel and Darwin worked without having any idea of what DNA was. Friedrich Miescher in 1869 discovered the substance called DNA in the nucleus of the cells. This stood for deoxyribonucleic acid. RNA, of course, is ribonucleic acid. And so the names of these nucleic acids is dependent on the sugar type. That's another reason those sugars matter so much. And so we really didn't know that this substance existed before 1869, let alone what we know about it now. In 1900, because Mendel didn't do very much ground truthing to disseminate his work, and it didn't really take off in the 1850s. It wasn't until 1900 that his laws were rediscovered. And it wasn't until 1909 that we even had a word for the genetic component, the molecular component that underlies a trait. So in 1909, we got the word gene from Johansen. Originally, it was pangene and then just shortened to gene. And that is for a unit of inheritance. That's the idea that there's something written somewhere. At the time, we didn't know in what molecular structure, but there was some molecular encoding that was related to a macroscopic phenotype. That was later found by Avery, McLeod, and McCarty, that it was DNA that housed that genetic material. It was DNA that was the material that encoded genes. And so, right at the end of the Second World War, it's pretty damning to see that the people who were running this horrible, atrocious war on race and genetic superiority didn't even know what genetic material was and what substance in cells produced it. And so pretty horrendous mismatch between science and global events at that time. A couple of years later, in 1952, Hershey and Chase show that DNA replication is semi-conservative. This is the idea that you have two rungs of DNA, and when a cell divides, those two rungs of DNA will separate from each other, and then the replication machinery will synthesize brand new DNA right alongside it. So you end up with two copies of the DNA, but half of that copy was in the parent cell and the other half of the copy is brand new. So we say semi-conservative. It conserved half of what the starting materials were. Just a year later, in 1953, James Watson, Francis Crick, and Maurice Wilkins published a paper demonstrated the double helical structure of DNA and the base pairings and their orientations in space. And this is not just 70 years ago, it is 70 years ago today, February 28th, the day scientists discovered the double helix. Okay, so this was the day that Watson and Crick barged into a pub outside Oxford and said that they had discovered the secret of life. So these are the culprits themselves: James Watson, who was then 25, and Francis Crick, who was about 12 years older at the time. They published the structure with information and insights from Maurice Wilkins and information they had obtained from Rosalind Franklin's lab notebooks. This was their original paper that was published a couple of months later on April 25th, 1953, The Molecular Structure of Nucleic Acids, a Structure for Deoxyribonucleic Acid. And you can see that famous double helical anti-parallel structure down here in their figure. If you want to listen to a nice 4-minute listen about this, NPR did a nice story that you can see from that QR code. Now, back to our timeline here. a little over 20 years later, Fred Sanger and Alan Coulson published a protocol for sequencing DNA. So prior to this, we knew the structure, but we didn't have a protocol for defining A, then C, then G, then T, then C, then A, then G, then C, then A. We didn't have a protocol for that. And so they figured this out. And then very quickly after that, the pace of research intensified. So in 1983, Kary Mullis invented a process called polymerase chain reaction, which allows you to amplify individual sequences of DNA using a similar protocol to the Sanger sequencing protocol. And by the way, it should be mentioned that, you know, Fred Sanger is the only person who's won the Nobel Prize twice for chemistry. Kary Mullis won the Nobel Prize as well. And so this is a very lucrative and important field that has hastened the human grasp of our own fate. And that only got better and better in the 1990s with Francis Arnold, another 2018 Nobel laureate in chemistry for performing directed evolution. And then, of course, in 2014, Jennifer Doudna and Emmanuel Charpentier invented the uses of CRISPR-Cas9 for making changes in real time. That is not between generations, making changes to a living organism's genetic sequences, again, of which there should be a lot of ethical forethought before doing an experiment like that, but it's an extremely powerful tool. And that brings us to today, 2023, 3 years after Doudna and Charpentier won their Nobel Prize. And it should be noted here that in the last 70 so years of molecular biology, women have done the lion's share of advancements here. And so we can't move forward in this field without recognizing that there have been disparities, gender disparities, chief among which is kind of the poster child for being left out due to sex, and that is Rosalind Franklin, who died at the age of 37. It was her work that ultimately solidified the inspiration for the insight into the helical structure of DNA. She took this, her grad student rather, took this famous photo 51 at King's College London in 1952. She used an X-ray crystallograph. This is an X-ray source here that funnels them into this funnel here, and there would be like a DNA crystal hanging here by this little pin. The X-rays would pass through the DNA crystal, and then it would, the ones that make it through would be measured by this detector here. So what you're seeing are dark spots where on the X-ray film, there wasn't a lot of electrons that made it, or excuse me, a lot of X-rays that made it through. Everything else is a little bit grayer, grayscale tones. That's principally because those X-rays were gobbled up by the DNA structure. They were absorbed by the DNA structure. But what we can see is that there's this helical structure right along here. It kind of forms this decussation, this X here. And that's because there's this repeated structure in the crystal of a DNA that continually absorbs electrons in a very regular pattern. And so that was one of the major insights that they had, in addition to some distances that we're going to get into here in a second. That's what ultimately led to our discovery of DNA's double helix. And then, of course, Rosalind Franklin, she ended up dying of ovarian cancer. There was a family history there, but it's very likely that the very tools that she used, X-ray crystallography, perhaps accelerated her diagnosis and subsequent death from cancer. And it is a credit to her that we now understand that the very thing that may have contributed to her death is something that we now have a lot more control over to save lives today. So one thing that we should understand is that the sequence of nucleic acid determines its higher order structure. Again, ribonucleic acid RNA is a pretty flexible molecule. So we're going to start with DNA here, which has a much more regular structure. Again, we've got these anti-parallel strands where A is bound to T via 2 hydrogen bonds, and G is bound to C via 3 hydrogen bonds, and they're running in opposite directions. So on this side, you have 5' to 3', and on this side you have 5' to 3' running in the opposite direction. So this is what we would call the primary structure, just the sequence of nucleotides that are bound to one another via a diphosphoro or phosphodiester bond. And of course, these we can assume are all deoxynucleotides. Secondary structure is when it wraps itself up into a helix like that. We can see that it kind of wraps around fishtails back and forth along its length. Now, the tertiary structure for DNA is relatively uninteresting, just twists and turns. We'll get into a little bit about how flexible those turns are here in a second, but the main driver of our interest for this is going to be the quaternary structure, or the interaction between DNA and other macromolecules, such as histones. So DNA will wrap around these histone proteins, wrap around, and then those histone proteins begin to associate themselves into a thicker macromolecule called chromatin fiber, and then it gets thicker and thicker and thicker until ultimately you're left with a chromosome. Okay, now each cell, at least specifically in humans, contains about 2 meters of DNA. Okay, 2 meters end to end of DNA. That's quite a feat. That's quite a lot of molecule to have just wrapping around, and it's not a very organized structure. And so what these histones and the chromatin and the chromosin structures allow the cells to do is to have kind of an organized structure, organized molecular structure, to be able to keep it all in one place and only open the bits of it that need to be open at any given time. And so we've some of the measurements that they knew in 1953 was that if you started here on the outside rung and moved around to the back and then moved around back to the front again, that's what we call one turn. And so in one turn of DNA, there's a distance of about 36 nanometers and about 10.5 nucleotides per turn. We also knew that the width of a single DNA strand is about 2 nanometers. And so if you wrap that 2 nanometer DNA around histones, you end up with a structure that's about 11 nanometers thick. If you condense those into chromatin fibers, you end up with something that's about 300 nanometers thick. That can further condense into about 700 nanometers until you ultimately end up at the chromosome, which is 1400 nanometers, 1400 nanometers. So while we increased thickness by 700-fold, if you took every chromosome and lined it up end to end, it'd only be about 200 nanometers. And so what that means is that our 2 meters of DNA has been shrunk down into a space of about 200 nanometers. So while we increase thickness by a process of 700%, by a factor of 700%, we decrease length by several million percent, 10 million percent specifically. Now let's focus on the flexibility of this molecule. Why is DNA helical? Well, as you can see from this cable, when you bend it, the outside bit is forced to stretch and the inside bit is forced to compress. And when you twist it up, those forces negate. So DNA adopts that same structure, and that helical structure avoids any kind of strain on the phosphate backbone, especially as it wraps around those packing molecules. The flexibility of the structure is dependent on its sequence, and this arises again from the relationship between AT and AU and GC pairs. They are different because the GC has 3 hydrogen bonds and the AT-AU has only 2 hydrogen bonds. GC bonds being a stronger, stiffer bond is a key fact of nucleic acid biochemistry. In this case, the implication is that a series of A's and T's will be more flexible than a series of G's and C's. It will also take less energy, less heat, that is, less temperature, to unwind this series of DNA, this A's and T's, as it would to unwind the series of G's and C's. We've kind of been building to this point now about the DNA protein interactions, because DNA on its own won't do anything. It is dependent on its protein counterparts to carry out its its instructions. And so we need DNA to be able to interact with proteins. And so there are many proteins out there that have molecular motifs that are designed to interact with the primary structure of DNA, okay? So we've got promoters and repressors that bind to specific sequences of DNA. Promoters will promote that sequence of DNA being active and actively transcribed, and repressors will prevent that sequence of DNA from being transcribed. We've got zinc finger nucleases, which, as you can see here, interact with the major groove of the protein, or excuse me, a major groove of the DNA, and recognize a specific sequence. And these nucleases are capable of chopping up DNA at specific spots. We've got a helix turn helix where you've got a groove here. The protein goes away from the DNA helix and then rejoins it at the end of another turn. So this is your full turn here. So you've got a alpha helix and then one whole turn and then another alpha helix. So helix turn helix. It's another molecular motif that proteins are going to use to interact with DNA. And then we've got leucine zippers that again is interacting with two major grooves of the DNA. And then we've got polymerases that we've talked about so far already that should be able to interact with the proteins or the nucleotides that they're going to be polymerizing. We've already talked about histones, but we're going to take a quick look at the molecular mechanism underlying that interaction. So here in ribbon format, we've got the histone protein, and then in the gray outside, we've got the DNA. If we zoom in on this interaction between them, we see that we've got the DNA, we've got the histone. The histone's going to have these lysine residues hanging off the end. And as we know from previous, earlier in the semester, lysine residues, when they carry that extra proton, which they would at physiological pH, are going to be positively charged. And we know that the backbone, the phosphodiester backbone of DNA, carries a negative charge as well. So these are going to be attracted to each other. We've got lysine residues attracted to the phosphate backbone. So there's going to be a nice tight association between the DNA and the histone. However, we don't always want that. DNA that is tightly packed is great for when you're packing things up and separating cells during cell division, but when the cell needs to go about its job of transcribing genes and translating proteins, we need to have access to some of these proteins that would otherwise be bound up by histones. So what we can do is we can take these histones and add an acetyl group to the end of them by acetyl-CoA. So there's enzymes that acetylate lysine, so they'd be called histone acetyl transferases, or HATs, and they would put this acetyl group on here, preventing that lysine from carrying a charge, and we would call that an acetyl-lysine residue. And so it has no charge, and it would have no affinity, no increased affinity for the negative backbone of DNA in that case. And if it was time to pack up the genome and these associations need to be stronger, we would rip that acetyl group off the lysine, off that acetyl-lysine via a histone deacetylase, or HDAC, and that would return it to the positively charged lysine that we would give us an attraction here. So this is how those two enzymes, histone acetyltransferases in the forward direction and histone deacetylases in the reverse direction. That's how cells control how much DNA is bound up by histones. So let's return here to our central dogma. So we have some major components to look at here. We've got DNA to RNA, RNA to protein. These are the arrows represent several processes: replication, transcription, and translation. Those are the processes of duplicating DNA sequences, creating RNA based on a DNA template, and creating protein based on an RNA template. And then, of course, reverse direction would be creating DNA from an RNA template. These are all carried out by enzymes. Replication is carried out by DNA polymerases. Transcription is carried out by RNA polymerases, and translation is carried out by ribosomes, which are essentially amino acid polymerases. So if we put this in kind of a table format here, we can look at the differences and some nomenclature for the names of the enzymes here. So in prokaryotes, the prominent primary replication polymerase for DNA is called DNA polymerase 3. That's a very powerful tool in molecular biology. In eukaryotes, it's more of a complex of 3 different proteins, DNA polymerases, alpha, delta, and epsilon. Each of these DNA polymerases come in repair flavors. So in prokaryotes, DNA polymerases 1 and 2 are the repair enzymes, whereas in eukaryotes, DNA polymerases beta, lambda, sigma, and mu are the repair enzymes for DNA. For transcription in prokaryotes, it's carried out by RNA polymerase, kind of an all-in-one jack of all trades, whereas in eukaryotes, it's more of a divide and conquer. So you have RNA polymerase 1 that generates the RNA that goes into the ribosome. RNA polymerase 2 transcribes DNA into mRNA that will then feed into a ribosome. And then RNA polymerase 3 generates the tRNAs, or the transfer RNAs, that are going to carry, they're basically molecular handles to carry amino acids into the ribosome to base pair onto the mRNA and give us a amino acid and grow an elongating chain of proteins. Or an amino or peptide rather. As far as ribosomes are concerned, prokaryotes have a 70S, whereas eukaryotes have an 80S. The S is a unit of weight. So essentially you take these ribosomes, you isolate the ribosomes from the cells, and you put them in a centrifuge in a certain kind of sucrose solution, and you just spin them around, and then as far as they get into that sucrose solution, it's like a literal physical distance that determines the S, or in this case, you can think of it as kind of like a molecular weight. They're broken down into 2 subunits. Prokaryotes have a 50S and a 30S, and yes, I know that doesn't add to 70, and 60S and 40S also do not add to 70, but when they're in a complex, they don't travel that much farther than the 60S, or proportionally farther than the 60S or the 50S. And so that's why those numbers don't quite add up. Just know that the main thing to understand from this slide is that prokaryotes have different replication, different transcription, and different translation machinery than do eukaryotes. And the differences between their replication, transcription, and translation machineries and ours are all differences that we can exploit when we're designing antibiotics to treat disease, okay? Some nomenclature to be aware of. DNA polymerases read DNA and they write DNA. Therefore, we call them DNA-dependent DNA polymerases. RNA polymerases read DNA and write RNA. Therefore, we call them DNA-dependent RNA polymerases. A reverse transcriptase would be an RNA polymer, technically it's a DNA polymerase. It's a RNA-dependent DNA polymerase. That would be a reverse transcriptase. And ribosomes read RNA and write amino acids. Therefore, for posterity's sake, we could call them RNA-dependent amino acid polymerases, even though I've never heard anyone say that, but just for continuity. Now, let's talk about a specific polymerase with kind of a mind of its own. So telomerases are a kind of polymerase that don't need their own template because they have it built into them. And so that's very interesting, and it's very helpful for our genetics. So we've got a picture here of chromosomes in yellow, and their ends are in blue. And those blue ends, they just suck up that blue dye because they are very rich in the same sequence over and over and over again. And those telomeres are basically kind of just a buffer at the end of those long chromosomes to protect what might be precious genetic material at the ends from getting frayed by DNA damage at the end. So those telomeres are very useful structures, and they were discovered, and the enzymes that create them were discovered by 3 Nobel laureates on the screen before us. We've got Barbara McClintock, who worked on corn, and then Carol Greider from, I believe, Australia. And then again, close to you, Elizabeth Blackburn works at the University of California, San Francisco, just a BART ride away from you. Telomerases are ribonucleoproteins with their own template RNA built into them. That is to say that they are proteins that have an RNA sequence built into them, and they use that RNA sequence as a template to build DNA. So it is a RNA dependent DNA polymerase. And so this is what I was foreshadowing earlier when I was saying that just a little bit of reverse transcription is needed to complete the replication process of the human genome, because it really depends on making these telomeres. So you replicate the whole genome, and then you add on the telomerase caps at the end, and that was made by a ribonucleoprotein. So let's talk now about why DNA is the reason you really should wear sunscreen. Okay, so let's say we've got a DNA chain here. It's DNA because it's missing this hydroxyl group at the 2', and then both of these are thymidine. Okay, so we've got a thymine group here and a thymine group here. They don't look exactly the same here because they're just a mirror image. This one was just rotated in the opposite directions. They're still the same molecule. Well, let's say that in the sun, these ultraviolet photons exist. One of them might happen to excite that double bond, which kicks some electrons over there, which kicks some electrons over there, and forms this deoxythymidine dimer, where you have these two bonds bound to each other. And that introduces a little bit of an angular strain on the DNA chain, whereas normally we had a nice kind of linear relationship. Now it's a little bit kinked, okay? This kink, it cannot be, it distorts the hydrogen bonding to the complementary base pairs on the other side. This would stall a DNA polymerase, so DNA repair enzymes would need to come in and chew this up. And the problem is when they chew it up, sometimes they repair it well, and sometimes they don't. And when they don't repair it well, that is going to be the case where, let's say they put an A where there should be a T, or they put a G where there should have been a T. That is where a mutation can happen. And so sun, ultraviolet photons, can cause thymidine dimers to exist, and then the repair process might make a mistake and might produce a mutation that, if in the wrong place, let's say in a cell cycle gate gene in the genome, might lead to unregulated cell cycles, unregulated replication and cell division, and that would be what we call cancer. There are a lot of other reasons to wear sunscreen. There's other mechanisms by which DNA is damaged by UV photons, and X-rays for that matter. But this is a pretty good start. So let's talk now about some practical applications of nucleic acid biochemistry. It always comes back to this question: how would you break a polymerase? The reason we might want to do that is because when you just give a polymerase a bunch of nucleotides and let it do its job, it will go through and it will create a whole new sequence of DNA, a whole new duplicate of the DNA without revealing any of its secrets. So if we can slow that down, we can slow that polymerase down, maybe we can see some of what's going on in the middle. And this is ultimately what Fred Sanger won one of his Nobel Prizes for. So this is deoxyadenosine triphosphate. This is what would be added to the growing chain as DNA is polymerizing. And one way that you can break a polymerase would be to stop it from adding anything to this 3' hydroxyl group. And so that might be what you have here with dideoxyadenosine triphosphate. So you've got your adenosine, you got your triphosphate, but you're missing both hydroxyl groups on the 2' and the 3' carbon. And so missing that 3' hydroxyl group, the dehydration reaction would not be able to proceed in the active site of the DNA polymerase. And so this is one way that you could break a polymerase using what we call a chain terminator, because you lose that hydroxyl, you lose the ability to grow the chain. So now let's talk about how that might look in what's called Sanger sequencing of DNA. So we've got our 5' to 3' sequence of DNA. We'll add a short primer that's complementary to the region here. It'll be the opposite direction, 5' to 3'. That primer will recruit DNA polymerase. It won't, DNA polymerase won't start replicating without a primer telling it where to start, and it won't be able to elongate without something, a 3' hydroxyl ahead of it to start adding things to. So let's say in this case, we give it everything it needs, all the deoxynucleotides, and it'll go along making this chain until it comes across, say, this deoxyguanosine molecule, and it's going to want to add a deoxycytidine to it. However, let's say by accident, the molecule randomly grabs a dideoxycytidine, and it can no longer elongate the chain past this. So what we have then is a situation where the fragment separates from the DNA, the template strand, the polymerase falls off, and the process begins again over and over again. And the idea here is that if you do this enough times, you will end up with fragments that are exactly the fragments that end at each individual base pair between the primer and the end of the 5' sequence that we're looking at. I guess if you do this enough, you let it run enough cycles, you'll end up with enough of these fragments that and each of these fragments is going to have a different weight. And so by separating them out by weight, we can see which came first. And we know when we know which came first, we can reconstruct what the sequence would have been of that DNA in the 5' to 3' direction based on what we learned by sequencing and synthesizing DNA using a 3' to 5' template. So that's what we call Sanger sequencing, and it's the basis for kind of gold standard sequencing that we know of today. Now, another major application of nucleic acid biochemistry is mRNA vaccines that we've all been accustomed to now with COVID. So you take mRNA solution, you inject it into the into a muscle, it's going to pump out all of these proteins via their ribosomes. And all of these proteins now are substrates for the immune system to come in, recognize this molecule, and then be prepared and ready to take on COVID. And that's where you get your immunity from. Now, there's still some room in nucleic acid biochemistry for us to improve these, because, like I've been saying so far, mRNA is a super unstable single-stranded molecule. And because of that, it degrades very, very easily. And so these mRNA vaccines need to be stored at super cold temperature, -80°C, for them to be transported and delivered safely into a person and to have some efficacy. Otherwise, if it degrades, then you're just pumping someone's muscle full of a bunch of nucleotides without a certain sequence. They're not going to do anything, okay? So nucleic acid biochemistry still has some way to go to figure out how to make mRNA vaccines stable at room temperature, and that would improve the accessibility of these molecules to be useful and more equitably distributed throughout the globe to places that don't have the same infrastructure as the United States. Let's move on now to another way that we can use nucleic acid biochemistry. So as I mentioned before, some of these molecules, these nucleosides, can act as neurotransmitters, and adenosine is one of them. If someone has an erratic heartbeat, the inhibitory effects of adenosine, if you inject it into the patient's blood, it will slow that person's heartbeat. Technically, it'll stop it for about, it'll take about 6 seconds, it'll stop it for a little bit, and then it'll come back online, the heart will come back online, and hopefully that person will have cardioverted out of that abnormal arrhythmia. Usually for something called SVT, supraventricular tachycardia. So adenosine is an inhibitory neurotransmitter. It's also an inhibitory neurotransmitter, not just in the heart, but in the brain. And so adenosine active in the brain will make you feel kind of sleepy and groggy because of the neural circuits that it's in. But caffeine, because of its structure, very, very similar to adenosine, it will sit in adenosine's seat, basically. It will, because it's such a similar structure, it will find the receptors of adenosine, it'll sit in those seats, and it won't really allow adenosine to act. And so we would call that a competitive antagonist, competitive inhibitor, rather, of the neurotransmitter adenosine. So if you inhibit an inhibitory neurotransmitter, then a negative plus a negative is a positive. And that's where caffeine gives us our feeling of alertness. Another major use of nucleotide and nucleoside biochemistry are antiretrovirals. So azithromycin is a nucleoside analog reverse transcriptase inhibitor, NRTI, discovered by two women, Marty St. Clair and Jade Rideout, in 1985. They were working very desperately at the height of the HIV/AIDS pandemic to come up with a molecule that would prevent HIV from replicating its RNA viral genome, or integrating that genome into the human genome. This chemical, azithromycin, it looks like thymidine, but it's essentially a chain terminator. Because of this azide group here instead of a hydroxyl group here, if a reverse transcriptase grabs a hold of this, it will not be able to elongate past this. And so for every adenine in the template genome of the HIV, it's going to be looking for a thymidine for every, it'll be looking for a uracil, because it'd be looking for, I apologize. It would be looking for a thymidine, because reverse transcriptases are RNA-dependent DNA polymerases. It'll be looking for a thymidine every time it sees an A while it's trying to replicate it. But because when this part would be, bound to a phosphate above it, or sitting in that spot, there'd be no space for this chain to elongate. And so very much like a competitive inhibitor that we talked about above with caffeine and adenosine, this will basically just sit in the reverse transcriptase active site where thymidine should be phosphate bonding to the previous nucleotide, and should be able to have an elongation of the chain after this. But because it's lacking the phosphate, and because it's lacking a 3' hydroxyl, neither of those are going to happen, and it will essentially stall the reverse transcription process. I want to make it very clear that before AZT, HIV was a death sentence. So nucleic acid biochemistry saves lives. Okay. Now, today, we've advanced our pharmacology of nucleoside analog reverse transcriptase inhibitors to the point where we're no longer just treating people who have HIV/AIDS, or where it's progressed to AIDS, but we are now prophylactically treating high-risk patients and demographics with these NRTIs. So emtricitabine is a deoxycytidine analog, but again, there's not even a 2', there's not even a 3' carbon. It's just been completely replaced with a sulfur group, and there's no way to add to this chain as well. There's no phosphate group as on the 5' carbon, so there's no way to incorporate that, although it probably would be phosphorylated in vivo in the patient. And so whoever in the Mentimeter quiz earlier said sulfur, no extra credit, but this is where that relates to throwing the sulfur into the nucleotides that we care about. This fluorine atom is very electronegative, and that just makes it stick in the reverse transcriptase enzyme a little bit better as well. So in combination with another nucleoside reverse transcriptase inhibitor called tenofovir, emtricitabine and tenofovir are used as pre-exposure prophylactics, such that patients who are receiving this treatment are essentially protected from contracting HIV. And if they have HIV, they're essentially protected from disseminating the virus. So this is where the phrase untransmittable, or excuse me, undetectable means untransmittable. Okay. So when we think about nucleosides and nucleotide biochemistry, nucleic acid biochemistry, I want to think about how the understanding of these molecules and how these bonds work together, how the hydrogen bonds work to each other, how the nitrogenous bases are bound to sugars, which are bound to phosphates. Understanding all of that biochemistry saves lives, okay? Knowledge is power. Understanding these molecules gives us power to save lives. So some things I want you to know, that you must know if you're going to be able to complete the assignments for the course. Big base, little name. Purines have 2 rings on them. That's a big nitrogenous base with the little name purine. Pyrimidines have 1 ring on them. That makes them a little base with a big name, okay? Purines always hydrogen bond to pyrimidines and vice versa. And then nitrogenous bases are bound to ribose or deoxyribose via glycosidic bonds, and that glycosidic bond is formed by a dehydration reaction. Nucleotides are formed polymers, that is, nucleotides bind to other nucleotides via phosphodiester bonds that were themselves also formed by dehydration reactions. Different machinery, but same mechanism. DNA is just RNA that's missing a 2' hydroxyl and uridine. The primary structure or the sequence of DNA influences its higher order structure and the proteins that it will interact with. The phosphate backbone will interact with histones no matter what, but the leucine zippers and the helix turn helix motifs, those are going to depend on interactions with the primary sequence, primary structure. You got those DNA and histones. Nucleic acids are read from 5' to 3'. You read the template from 5' to 3', but you are synthesizing along the way in the 5' to 3' direction. Sorry, so nucleic acids are read from 3' to 5', but they are synthesized 5' to 3'. They run in opposite directions. And then, of course, DNA denatures with increased temperature, and this is absolutely necessary for PCR and Sanger sequencing and much of the technology that we use today. So let's go ahead and return to Menti, and then go ahead and fire off and let me know what you have on your mind. Okay, difference between euchromatin and